Thank you. Thank you. Welcome back, everybody. Okay, so today we're going to be continuing our walk through the GI system, continuing thinking about our process of absorption. So when we left off, I mentioned we start digestion in the mouth. So you're chewing food, mastication. That's physically breaking down food. When we say the word digestion, we mean breakdown of food ultimately into monomers of our various macromolecules that we can absorb across our intestinal lining. So we started with our carbohydrates. So that especially we want to think of as starting in the mouth, as we've got salivary amylase that's responsible for kicking off our digestion of starches in particular. But we know that's not the only thing. We're also sending those carbs down through your pharynx, esophagus, stomach, into the small intestine and the duodenum, where they encounter our pancreatic juice coming in, including pancreatic amylase, which continues that carbohydrate digestion. That gets us to disaccharides and limit dextrins. So these are what we were able to break that starch into. But we do need to get to a monosaccharide in order to fully absorb into the intestine. So I pulled out an example of one of the final ways that we managed to break down those disaccharides and limit dextrins. So we got lactase, super important enzyme. If you're lactose intolerant, this is probably what you're missing, right? So this is a brush border enzyme, and its job is breaking down lactose, which happens to be a disaccharide. So that's those two monomers stuck together. So we can't directly absorb lactose, which is kind of our problem. Okay. So we got this brush border enzyme lactase that can break it down. So where are we going to find that based on the information we have here? Okay, let's take a look. Oops, did I say it out loud? I totally did. Okay. Well, the way you would get to this, it's going to be on the apical membrane of your GI cells, going to be in the small intestine where our absorption is really happening in that first 20%. The way you would be able to answer this question, if I hadn't said it out loud accidentally, right, we notice this phrase brush border enzyme. Okay. So the brush border, remember, is a word for when we have cells that have a lot of microvilli on them. So that kind of fuzzy look to the lining of that cell layer is an adaptation for absorption, gives you lots of surface area, lots of room for molecules to bump into enzymes embedded on that brush border and get absorbed or get broken down and then absorbed. So lactase is an example of one of the types of enzymes we would have in that brush border, bless you, fully getting us down to those monosaccharides that you're able to absorb across a cell membrane. Okay. All right. So we had left off with our proteins. We mentioned these three classes of proteases that we're going to be paying attention to. Our endopeptidases that split interior peptide bonds, our exopeptidases that cleave amino acids off the end of a polypeptide, and our zymogens. Okay, so of those that we just mentioned on that last slide and that we spoke about at the end of Monday, which one was an inactive form of a protease? Okay, take a look for everybody playing along. Yeah, it's the zymogens. So the exopeptidases and endopeptidases are both already functional. They're ready to go. They can break polypeptides. They just have certain spots in our polypeptide chain that they're physically like able to get to. But the zymogens, we still have to activate, right? So that's that bit here, right? So we're going to need to do proteolysis, so protein breakdown, in order to break these zymogens down a little bit, which is how we convert them into their active form. Okay. Cool. All right. So we're going to track our way through how you break down a protein, right? So say you eat a burger, right? We dealt with our bun and our fries, our carbohydrates. Now we're going to think about the protein part itself. Okay. So as you chew up your food, not much is going to happen to the protein as it's in your mouth, right? You just manually kind of start to break it up, swallow it down your pharynx, esophagus, and then you get to the stomach, which is where the actual chemical digestion process is starting. Okay, so what we want to remember here, what we're looking at is our gastric pit here, right? And remember, our gastric pit has both our chief cells and our parietal cells, right? Chief cells and parietal cells in this gastric pit, in these glands at the base, right? And one of the things that we're doing here in this gastric pit is secreting an enzyme, essentially, that enables us to start this protein digestion process, right? So we had our chief cells secreting our pepsinogen, right? So we got a chief cell secreting pepsinogen. We can see that here, right? Pepsinogen coming from our chief cells. Okay. And the other thing that we're producing down here in our gastric glands in the gastric pit is we're producing acid, right? So we talked about the hydrogen ions in particular that we're producing, right? So what we're going to end up with basically is hydrochloric acid coming from our parietal cells. That's the kind of full form of this acid. Okay. So pepsinogen, which is inactive from the chief cells, very strong acid, hydrochloric acid coming from those parietal cells. Okay. So if we want to digest a protein, we need an actual enzyme that's able to digest our protein. Okay. And we've got this inactive form, the pepsinogen. So actually the first thing that we're going to have to do is break down pepsinogen so that we have pepsin, an active enzyme. Okay. Now, fortunately, the other side of this gastric pit producing our hydrochloric acid, the strong acid, lets us do that because of this process or this denaturing process, right? So this high concentration of acid, this really low pH, our stomach pH of 2, means we're unfolding proteins, messing with their shape just based on this super acidic environment from that hydrochloric acid. So that's what activates the pepsinogen, right? So the pepsinogen itself gets activated by that acid into pepsin, okay? And now we've got pepsin in our gastric juices in the stomach, ready to break down other proteins. So like the pepsinogen, other proteins would have started to be unfolded. So what we're looking at here is a protein that's been uncoiled, unspooled, and we see this long polypeptide chain. All right. Okay. So we're now going to have pepsin come in, and it's going to help us start to break that apart. All right. So we can see we broke off a piece there. All right. We broke another piece off here, something like this, right? So now we have our smaller peptide fragments after pepsin is done doing its job. So we're not all the way down to a small enough size that we're going to be able to absorb these. We're going to have to keep breaking down this protein, but we're partway there, even as we have our proteins getting sloshed around with the gastric juices in this chyme fluid that we have in the stomach. Okay. So we are going to then see that chyme with those peptide fragments and squeeze through that pyloric sphincter into the duodenum, which is our first part of the small intestine. Okay. So this process is now going to continue. We've got partially digested proteins in the duodenum, And also coming into the duodenum, we have our ducts, right? So you're going to have pancreatic duct, and also our common bile duct is going to be coming in here, but we don't need to think about that one for proteins, all right? We're going to be focusing on the pancreatic duct for right now. Okay. So we've got our partially digested protein, we've got our pancreatic juice coming in through this pancreatic duct, carrying all its enzymes, right? So for the carbohydrates, we had our pancreatic amylase in that pancreatic juice. Now we've also got a lot of proteases to add to our mental list of all these different enzymes and proteins the pancreas has been producing. Okay. So here we have the proteases coming from the pancreas. So we've got trypsin, chymotrypsin, and carboxypeptidase all coming through that pancreatic duct in our pancreatic juice. And actually, you might notice on your figure here, right? These are slightly different words than in your bullet points, right? We see we have trypsin, ogen, chymotrypsin, ogen, and procarboxypeptidase. What that tells us is that in this initial form, as we first get to the lumen of the small intestine, these proteases are in kind of zymogen form. They're in their inactive form. Like our pepsinogen, they're going to need to be activated. Not hard to activate them once you're in the small intestine. That happens pretty quickly. But if you notice that difference, that's why. Also in the small intestine, we've got enzymes in our brush border. So that lining of the duodenum, the lining of the small intestine, has enzymes embedded in or on it. Okay? So we can see an example of one of those here. So we see this enterokinase, right? So one of the things that that enterokinase does, you can see, takes that trypsinogen coming in our pancreatic juice. When trypsinogen bumps into the wall of the small intestine, runs into this enterokinase, and now we can see that when it gets released by enterokinase, it's in this active form trypsin. And then we can see trypsin swimming over and activating our other two proteases here, right? So this is how we get to that fully active form of all these enzymes that are helping us continue our protein digestion. We have other brush border proteases. Aminopeptidase isn't involved in this activation process in the same way, but it would look the same as this enterokinase basically if it were on this figure. So it'd be something we're waiting for proteins to kind of bump into the wall here to continue digestion. Okay. So as we do that, right, these different proteases can then break down our peptide fragments even smaller, right? And our goal is to get down to, say, a dipeptide, tripeptide size, right? That's going to be kind of the biggest we're going to be able to transport across a cell, right? So dipeptides and tripeptides, depending on which ones they are, we're going to have specific transporters or carrier proteins for those specific di and tripeptides, right? And they're going to be able to actively transport those peptide fragments, right? If we have a single amino acid, it's even easier, right? So if we manage to break it down to just one single amino acid, then we are going to use our ATP pump to get some energy, right? I got some energy, or use some energy to make the gradient, I should say, right? And then we can do secondary active transport like we did with glucose and focused in on in the kidneys, right? As we were talking about filtration. So we now know how we can use a sodium gradient that we build up in order to do secondary active transport. So some of the smallest bits, the amino acids, are going into essentially your bloodstream, right? Because we cross this absorptive layer, right? We cross through the inside layers of our small intestine, and eventually our goal is to get to the bloodstream, right? So we're on our way there. with facilitated diffusion or active transport for specific di and tripeptides. Okay. Once we are inside a cell, even if we picked one of these larger parts, the di or tripeptides, which is just 2 or 3 amino acids, we would then break it down to just a single amino acid inside the cell before we pass it on to the blood, basically. Okay. I'll just walk us through in order, just so we know. Okay, so we chewed our protein, didn't chemically digest it in the mouth, swallowed it, hits the stomach, denatures, which means unfolding, start breaking it up with pepsin. So we're breaking it into some peptide fragments, some chunks, head into the duodenum, the first part of our small intestine, where we encounter our pancreatic proteases that break us down into 1, 2, or 3 amino acids together. We got ways of actively absorbing those into cells. If we picked a di or tripeptide, we break it down to a single amino acid once we're in the cell, and then we can pass that towards the bloodstream. Is our like high view overview of what's going on with the proteins. Good. So we're now going to move on to our final big molecule that we're thinking about absorbing, our lipids or our fats. So to understand why our lipid digestion, bless you, is a little bit different, we want to remember the properties of a lipid, right? As they're really their chemical properties, I would say, are going to change how we have to digest and then absorb them. Okay, let's take a look. Okay, yeah. So we have all kinds of lipids in our bodies, in the world. They can look sort of different if you are looking at the chemical structure of one, but what's going to unite them as a group, unite them as a macromolecule class, is that they have substantial portions that are nonpolar, right? They're nonpolar because they're essentially mostly just hydrocarbon chains. So carbon and hydrogen don't have a lot of difference in electronegativity. They don't hog electrons. We don't have little charges building up, right? So that's the nonpolar piece. And they're hydrophobic, so they run away from water, right? Oil and water. Because of this nonpolar property, water is a polar molecule. It does have these little dipole moments on it, these little charges, mini charges, right? So we have this kind of clash between water that's looking to kind of share little bits of charges, little bits of electrons, and our lipids that aren't. They're stable as they are. Okay. So any lipid, we're going to have a large portion of it that's nonpolar, hydrophobic, running away from the water. Okay. We do have some special lipids that are amphipathic, and they're going to come into play here as well. But when we're thinking about the ones we're digesting, it's mostly not that. Okay. So your typical diet should include about 50 grams per day, let's say, of lipids. Almost all of that is going to be in the form of triglycerides. So remember, a triglyceride, I always think it looks a little bit like a flag. We got our glycerol backbone, and then we've got our 3 fatty acid chains coming off the end. Right? We've got saturated, we've got unsaturated, we've got trans, we got cis, but this is the basic form. Okay. That's 90% of your lipids are these fats and oils, these triglycerides. Okay. So you eat a fat or an oil, you got a little bit of a problem as you're going to digest and absorb it, right? Because we have this hydrophobic property, which means that our lipids are not water soluble, right? If you mix some oil into water, you're just going to end up with a big bubble of oil or a big layer of oil sitting on top. We're not mixing into the water really at all. Okay. This means that as you eat lipids, they're not really going to mix in with the stomach contents. They're not really going to mix in with the intestinal contents. They're just all going to try to group together as one big, basically fat glob, right? Now we're forming kind of a fatberg as we go into the stomach and the intestines. We can't just absorb a giant fat glob through a cell. That's not going to work. We do still have to chemically break down these lipids. So we're going to need to find a way to break up this big globule of fat into smaller fat droplets and then be able to actually attack these lipids with an enzyme. So our process of lipid digestion is going to involve a bunch of different organs and kind of partners that are letting us do all these parts of the process. So ultimately, when we go to chemically break down a triglyceride, right, pull off fatty acid chains, basically, we're going to use a lipase. So ascending tells you it's an enzyme. Lipe, that first part, tells you we're breaking down lipids. And these are going to come from our pancreas, right? So just like we have our pancreatic amylase for starches, we've got our proteases for proteins. Your pancreas is also making lipases to break down lipids. Okay. But the lipases can only break down a molecule that they can actually come into contact with. So if you've got a big fat droplet, the only thing you're going to be able to get to is the very outside, which means you'll have a ton of fat still in the middle that this enzyme can do absolutely nothing about. So this is where bile comes in. So we know that we're making bile in the liver. We're storing some extra bile in the gallbladder, and eventually it comes in through the common bile duct into that same spot where we have our pancreatic duct joining with it to put these combined digestive enzymes and molecules into our duodenum, right? So in the bile, in addition to having the kind of waste products from breaking down hemoglobin, we also have these things called bile salts, okay? So our bile salts are going to help us break up our fat droplets. So we're going to try to get as much surface area as we can for our fats because it's that surface that our lipases from the pancreas can get to. So our bile salts are emulsifying these larger fat droplets into smaller ones, is the way we would say it. So this process of emulsification means that we're making fat able to mix in with this watery environment that we find in the small intestine. Okay, so here we're looking at what a bile salt looks like. Okay, so a bile salt is made in the liver. It's made from cholesterol, which itself is a lipid. It's just not a triglyceride, it's a steroid, right? Okay, so we can see that sort of steroid base here, right? We look for these kind of four rings pressed together. That was how we would recognize a steroid if we were just trying to tell what class of macromolecule we're looking at. So that's what we're making our bile salt from. And functionally, though, the salt part gives a little bit of an extra function. So bile salts actually are going to be amphipathic, like our phospholipids were. That was our other type of lipid that we really thought of as having these dual properties of having a hydrophobic side like any other lipid, right? So we've got this non-polar part to them, okay? But we also have this hydrophilic side, right? So this bile salt here, made from cholesterol, but we've added stuff, means that we now have a part of this molecule that's going to be attracted to water, that hydrophilic side with some polar groups. But we also have a side that's going to mix in nicely with our fat droplet, be able to come into contact with it. So as we secrete these bile salts and put them into our small intestine, these bile salts are going to be able to emulsify our fat as they kind of mix in. Right? So at the bottom, we can kind of see what they're doing. So looking at the figure, we started with a big fat globule. It's totally nonpolar. Everything is running away from the water. But then we have these bile salts come in from the liver. They're amphipathic. So they don't mind touching the water with that polar side, but also touching our triglycerides and our other lipids with that hydrophobic side, the part that's attracted to lipids. Right? So as they mix into the fat globule, they naturally start to break it apart, with the bile salts kind of sitting along the outside of it. So we can see that here. So here we have our smaller kind of fat droplets. Okay. So now once we have these smaller fat droplets, we're at a point where we can actually start to break apart our triglycerides. Okay. So that is the job of the pancreatic lipases, ascending enzyme, right? So our actual enzymes are going to start to attack here, right? So they're going to come in to get our triglycerides, and they're going to start to chop them apart, basically, which we'll see on the next slide. So when we pull apart a triglyceride, we have those three parts. We're going to pull basically these two fatty acids at either end off, right? So we're going to be pulling off a fatty acid chain, a fatty acid chain. We'll be left kind of like with this T in the middle usually. Okay. So some of our fatty acids that we've pulled off and monoglycerides, so that remaining kind of chunk, because we also have this glycerol backbone, are going to be able to be absorbed at this point. Right? And because these are fatty acids, because they're nonpolar, they don't need help getting across the cell membrane, right? They can just squeeze between our phospholipids, right? So they're able to just diffuse across into a cell. Okay. Not every single fatty acid and monoglyceride is going to do that though, because if you've got a lot of them, we're basically trying to come to equilibrium, right? So even concentrations kind of across the lumen versus the inside of our cells, right? So some of these fatty acids and monoglycerides are going to stay in the lumen in the form of micelles, and then kind of slowly, one by one, little pieces are going to get absorbed. So we see that here. Okay. So what we're looking at, right, here is what we had from the previous slide. So we had our fat droplet with the bile salts on the outside, triglycerides inside, plus some other things, right? You ate something that had cells, so you're going to have some phospholipids. You're going to have some other types of lipids in here as well. Then we have our lipase from the pancreas coming in. So we can see that pulled out these triglycerides, and we have our monoglycerides and our fatty acids hanging out. Some of them just directly diffused into our absorptive cells, and some of them are still hanging out waiting in these micelles. So micelle, just a reminder, it's a natural kind of shape that forms with lipids, right? So it's like the alternate version of our cell membrane made of phospholipids, right? Our phospholipid bilayer formed because we have phospholipids facing each other and making a bubble, water inside, water outside. Our micelles, you can see these non-polar parts, these fatty acid tails, are all facing towards the center. So we just don't have a double layer. We don't have water inside. We just got this little glob of small lipids. Okay. So we're going to absorb dribble by dribble little bits of these fats into our absorptive cells. And once they're in there, we're going to do some processing. Okay. So we're actually going to start to rebuild those triglycerides immediately, right? We just needed them to be small enough to diffuse across our cell membrane. But once they're in the cell, we make them big again. We can do that in our smooth endoplasmic reticulum, so one of our types of organelles. And another organelle, our Golgi apparatus, we're going to make them into chylomicrons. So a chylomicron is a type of lipoprotein. So we're going to take these lipids and put a little protein in there too. And that is another form of lipid that we're going to be sending back to the body, but we're actually not sending it directly to the blood. All right, so we're going to be sending our chylomicrons first into the interstitial fluid, but then they're going to go in the lymphatic system instead of into a vein. Okay, so that is why we paid a little bit of attention to our villi, the big ones, not the microvilli, but the large projections that we have in the small intestine, where we've got arterioles, venules, capillary beds in this little projection, and a lacteal in the middle. Lacteal is like a branch of the lymphatic system. We need that for this fat absorption. Okay. So here we see our small lipids, the monoglycerides and fatty acids, just diffusing into our cell. And here we have an image of rebuilding a triglyceride. All right. And then we will also combine a bunch of lipids, a little bit of protein in the Golgi apparatus, and package this all into a chylomicron. Okay. As you can see, that chylomicron is in a little bubble, right? So it's in a vesicle. That vesicle comes up to our basolateral membrane. When it bumps into it, pops like a bubble, and the chylomicron bursts out into our interstitial fluid here, right? This is just the process of exocytosis. This is how you get something big out of a cell. And then we have that chylomicron skipping over the blood and instead getting absorbed into a lacteal, into our lymphatic system. And it will ultimately get back to your blood. Your lymphatic system does eventually dump into your veins. So you might have seen that with the thoracic duct in the chest. So dumping back in to the veins right before they hit the heart, right? But we're going to get carried around through the lymphatic system right until we get there. Okay. The other thing that we want to be aware of as we are thinking about this process of fat digestion and absorption is that those bile salts are reusable. Your liver doesn't have to remake them every single time. So we also have a process of reabsorbing those bile salts themselves. So here you can see the bile salts being produced by the liver. So we see them going into the hepatic duct, meeting up with the gallbladder, might have stored some, but it's not making any on its own. Bile salts in the duodenum, they do their job. And they're going to travel through, right? So they go through the duodenum, jejunum, and by the time they get to the ileum, they're getting reabsorbed and recycled, going into our portal system, to our hepatic portal vein, back to the liver, so we can use them again. Okay. So those are our three big classes of macromolecules that we want to make sure we know everything about how they get digested and absorbed. Okay, so those are our first priority when we're thinking about understanding nutrition and absorbing the contents of our food. But we do have a lot of other things that, from your body's perspective, are also important to absorb. We're just going to focus on them a little less. So one thing is you do need vitamins. You don't need as much quantity of vitamin as you do of carbs and proteins and lipids to fuel your body on a day-to-day basis. But if you don't get all your vitamins, you're going to be vitamin deficient. You're going to have a bad time. You're going to have cell processes that stop working without these vitamins. Vitamins basically come in kind of two big groups. So you're going to have fat-soluble vitamins. So if we think about fat-soluble, right, things that are fat-soluble are really going to be like non-polar, hydrophobic, right? We're sorting these two things based on what they mix in with best. So the fat-soluble vitamins, vitamins A, D, E, and K, and they're going to get absorbed with the lipids. So you're going to see them in your big fat globule. You're going to see them in the smaller fat droplet. You're going to sometimes see them in the micelles, and then they're sometimes going to get mixed into those chylomicrons that your Golgi apparatus is putting together. So fat-soluble vitamins, just following that lipid process. Water-soluble vitamins can swim around in your intestinal contents on their own. They're going to require special transport proteins for each vitamin. We're not going to go into the specifics of each of those special transport proteins, but we do want to mention one separate from the others, which is vitamin B12. So vitamin B12 is important for making stuff, particularly important for making hemoglobin. And the reason we're picking it out as one thing to single out is we mentioned intrinsic factor. Intrinsic factor was one of the other things that we're making in the stomach in those gastric pits. So that intrinsic factor is important for binding to the vitamin B12, which is the shape it needs to be in for your body to absorb it. Okay, so we want to absorb vitamins, both the fat-soluble ones, just with lipids, water-soluble ones with their own processes. We also want to absorb minerals. When we say minerals, I mean like electrolytes, salt, we mean ions, right? At least for our purposes. So we're going to be absorbing sodium chloride and potassium in particular. So sodium at first is going to get dragged along a little bit with water reabsorption. A lot of water is going to get reabsorbed paracellularly, so kind of like along the sides of our cells. Some of the sodium is going to be coming with it. And then we're going to have active processes for sodium absorption in the jejunum, in the ileum, and in the colon. So we're going to wait till we pass the duodenum, although we know that all our cells are using sodium potassium pumps for setting up gradients. And then we're going to actively be making sure we get as much salt as we need in these latter parts of the small intestine and into the colon. Chloride, because it has that opposite charge to sodium, sodium positive, chloride negative, it's going to passively end up following that sodium absorption. So we don't need to separately actively absorb the chloride because sodium is going to attract it on its own. And then we have potassium. So potassium is going to get passively absorbed. So not a particularly complicated process here. The one thing to know about potassium is that in certain conditions, right? So if it's super low potassium in the luminal concentration, right? Like, so the concentration of potassium is really low there. Then it's actually going to flip what it does. So this potassium, instead of being passively absorbed, if we have really low potassium in the lumen, will flip directions and end up getting secreted because it's just following gradients here. Okay. Another thing that we want to make sure that we track kind of separately here is bicarbonate, right? So we mentioned bicarbonate is super important to your body because it's a buffer for pH in your bloodstream, right? So in your circulation. So we need to make sure that we have enough bicarbonate to be able to keep the pH of our blood stable. One of the sources, hopefully the main source of that bicarbonate, is your diet. So you're eating something that has bicarbonate in it. It's going all the way through your digestive tract until it gets to the jejunum. So in the jejunum, we're going to start absorbing some of our bicarbonate ions, passively absorbing them. And then when we get to the ileum and the colon, we're going to actually end up secreting a little bit of bicarbonate. So the reason for that is to kind of continue the buffering of the contents of your GI tract itself. Remember, we have various fluids where we have bicarbonate in our intestinal contents, sort of because we're trying to neutralize that super strong stomach acid, right? Where we've got our stomach acid at a pH of 2, right? So we got really acidic stuff going into the duodenum. So there are places throughout your GI tract where bicarbonate will be kind of used right there to make sure you're not burning your intestines either with that acidity. Okay. Finally, calcium. Calcium gets absorbed in the duodenum and the jejunum, so those earlier parts of the small intestine. We've got a brush border protein that transports calcium. So make sure you get enough calcium. We've got this brush border protein on one side of the cell, a pump on the other side to make sure that that calcium gets all the way to your blood. We've mentioned iron before. We talked about why we need iron when we talked about the heme groups in hemoglobin. So we've mentioned these two words before. Right? We know a little bit about transferrin and ferritin. Right? So this is a reminder that we need to absorb iron. When we absorb iron, to move iron around, you need to bind it to transferrin to give us that T iron. Right? So we've got this T iron is how we had it listed on our slides when we were talking about red blood cells, right? So that transferrin part is coming from our enterocytes, the small intestine. It's basically the lining of the small intestine. The transferrin binds to the iron, and then we can absorb that complex through endocytosis. So is through the opposite of exocytosis, right? So we're making a big bubble, but pulling it into the cell. We store some of the transferrin in our enterocytes. So we kind of keep some of them in the lining of the small intestine for a while. When we're storing iron, we're storing it in this F iron form of ferritin. And then some of it is continuing into the blood, right? So we're going to have transferrin in the blood as well. And that's how iron will travel around and hopefully get to especially our bone marrow so that we can build heme for our hemoglobin. And we're not thinking about specifics of water absorption here, but because we focused on a lot of how we adjust water when we talked about our renal system and the kidney, a reminder, you are taking water in from the things that you eat and drink, right? So you're going to have water in the GI tract. Overall, hopefully we're having an intake of water, but we have a lot of water in our secretions as well. So you're hopefully like drinking from your mouth, like 2 liters per day of water. But with the pancreatic juice, our stomach acid, the bile, all that stuff, you're also going to be secreting about 7 liters of water into your GI tract. So we want to absorb both that intake and these secretions kind of later on. This happens passively. So we're just following our absorption of solutes. So osmosis, we're just following that kind of osmotic pull. And this is largely happening kind of in the colon. So we mentioned that the colon is kind of condensing everything, compacting our feces down. Okay. We're going to move forward into the very beginning of understanding how we're controlling the GI system, a little bit about secretion, a little bit about movement. So that's what we mean by motility. We mean movement of our GI tract. Okay. So we're going to start by thinking about some pathways. Okay. So when you're controlling your GI tract and all this GI function, right, you know, think about, okay, how do we know that the pancreas should make a bunch of proteases right now, right? Like, how do we know we need to secrete our bile salts now to emulsify our fats, right? We don't want to just be making tons and tons of enzymes all the time and just constantly dumping them into your intestines, because that's a waste, right? It's a waste of energy and it's a waste of time, right? So your body's going to try to predict, okay, when do we want these enzymes? Where do they need to be kind of at this time point? And that's the point of these different phases of GI control, or thinking about where is the stimulus coming from that tells us what to do. So we start thinking about this with a cephalic phase. So whenever you see this ceph beginning to a word, that's a Greek root that means head. So cephalic phase is coming from your head. Okay. So this means that your thoughts influence how your body starts to prepare to eat food and digest it. When you think about something like a food you're craving, right, that actually has physiological consequences. When you start to taste something, right, you don't have macromolecules in your duodenum yet to break down to the protease, but your body starts to know something's coming, right? When you smell food, your body's going to start to prepare. So we're going to think about this as having a bunch of reflexes that ultimately are going to have to involve the central nervous system, because that's where your brain is. So we're going to have these long reflexes that help us prepare for this digestive process, make sure our body's ready to digest and then absorb this food when it gets there. We're also going to have some more local stimuli to the food itself, right? So our gastric phase is going to be talking about all the stimuli that are coming from the stomach itself, right? So your stomach is going to be able to feel like, do I have stuff in my stomach or is it empty, right? And that's going to have an influence on how your GI tract is functioning. So we're going to have some shorter reflexes and kind of local stuff, and we're going to have some long reflexes with longer pathways. We're going to involve GI hormones here as well when we think about the gastric phase. Our intestinal phase Again, we're going to have long and short reflexes and GI hormones involved, and we'll separate out next time, like what those reflexes and stuff mean and what they are. But intestinal phase for now, we just want to know we got stimuli coming from our small intestine, right? So we're going to have a signal from our body. Okay, the chyme from the stomach is now getting squeezed into the duodenum. We now have food actually coming into contact with the cells that should start absorbing it. So that's going to trigger its own responses as we kind of move through our GI tract and try to get all these glands and ducts and organ systems working together tightly to produce the right things. So next time, we'll be thinking about the functions of our saliva, stomach acid, pancreatic juice, and bile from this perspective. Like, how do we know to release them? How do we know what should go in them at that specific moment?